Okay, cell division. But first, first, we talked about this before, but cells have to divide. Because that's how things grow. That's how organisms grow. And renew themselves. That's how organisms grow and renew themselves. We were on chapter 5. On chapter 5. And this is cell division. It's really not a bad chapter. It's pretty straightforward. So, for example, I think I mentioned this early on in the year. Every couple of weeks, all your skin cells are replaced. Every couple of weeks. So this is just an example. All your skin cells are replaced. Now, how else would that happen? The cells have to divide because then some of them die and get rubbed off and washed off and stuff, and new ones are made to replace it. And the way that this happens is that cells divide, okay? So basically you have what's called the parent cell. And the parent cell splits into two daughter cells. That way you have more cells, and now some of them can die, and you still end up with the same total number, okay? So that is the reason for cell division. Now, you might ask, why can't we just be like one giant cell? Well, for one thing, if you were one giant cell, it'd be kind of hard to replace parts of yourself, maybe. I don't know. But we also talked about how things move through cells only by diffusion. And diffusion is really slow. And so for something to diffuse all the way through liquid to get to the middle of your body, if you were just one giant cell, would take way too long, and you wouldn't be able to move very quickly. So if you were one big blob, one big giant cell, you would be just really slow. You'd have a really slow metabolism, if it was possible for you to live at all. So they have to divide. But here's the thing. They have to make exact copies. So these daughters have to be exact copies. Otherwise, you're going to be constantly like changing. Right? You might have like ears sprouting out of your hand or something like that over time. In other words, you want to replace a skin cell with a skin cell, not a blood cell or a bone, bone marrow cell or cell from your eye or something like that. So they have to be exact copies. Now, what that means is with copied DNA. And this is where the focus is, okay, for this chapter. Actually, remember, mitochondria, they copy themselves. I don't know if you remember that. Mitochondria reproduce themselves. So before the cell division takes place, one of the things that happens is the mitochondrion reproduce so that there's twice as many. And then half of them go to one cell and half of them go to the other. The other organelles are going to be built by the new cells as well. But the main thing we're going to focus on is the copying the DNA. Now, a parent. That's called the parent cell, and then when it divides into daughter cells. A strand of DNA with protein. Now, what is the protein doing? It's kind of like a packaging for the DNA. We talked in the beginning of the year about how genes can be turned on or off. That happens when there's like molecules of protein that cover up certain parts of the DNA. So the reason I put protein in there is because that's what makes what's called chromatin, which you've heard of before, DNA and protein. The protein, though, it turns out, as people have been discovering the last 10 years or so, actually plays a very important role. So it's something we should not forget about. Proteins are in there. They're turning on certain parts of the DNA, turning off other parts, making it coil a certain way, stuff like that. So a strand of DNA with protein is called a chromosome. Now, we call it chromatin if we're just talking about the substance itself. So you would say it's made of chromatin, but it's a chromosome. So a chromosome is made of chromatin, which is DNA and protein. Okay? Let me just write that here because it's kind of confusing. A chromosome, a chromosome is made, and yes, scientists do intentionally try to confuse you. That's why they make all the words sound the same. It's made of chromatin, which is DNA and protein. Okay, each of your body cells now, I say body cells because I'm not including sex cells. Sperm cells and egg cells don't work exactly the same way. Each of your body cells has 23 chromosomes. Well, the sex cells do also, but they only have one copy of each. Each of your body cells has 23 chromosomes. You might say it looks something like this. There's 23 strands. Okay, basically, if you had all these strands of DNA and you lined them up, now, this isn't really how it would look though, because they're long. They're not as short as this, unless when they're all coiled up. That's when they look short. But normally there would be a really long strand, like spaghetti. Longer than that. Each body cell has 23. Here's the thing. If your cells are going to divide and make copies of themselves, they have to copy all of those. All of these must be copied. And then, think about this. It's not enough just to copy them. You have to make sure that each new cell gets the full set. The same set. You can't just copy all of these and then just randomly divide them in half and give half to one cell and half to the other. Then the cells would be different. They have to have exactly the same 23. So you have to make copies, and then you have to literally pass one copy to this cell, one copy to this cell of each. Okay? So, for cell division, the cell has to copy all 23. Has to copy all 23 and make sure each daughter gets a full set. Otherwise, like I said, if a skin cell is missing the chromosome that has the gene for a certain protein, then it's not going to work right. It's not going to be a skin cell. So they have to have the full set. Make sure each daughter cell has a full set of 23. Now, what this tells you is that this means there's time in every cell when there are actually 46 strands of DNA. Okay? So, there will be a time when a cell has 46 chromosomes. Chromosomes. Two copies of a set of 23. There's a basic set of 23 different DNA strands. Now, by the way, all these strands, they're not the same, right? One of those strands of DNA up there might have the gene for eye color on it, and another one might have the gene for hair color, and another one might have the gene that makes your nose, and another one might have the gene that makes a proton-pumping ATPase enzyme. So if you were missing any of them, it's going to cause big problems. In fact, we're going to talk later this spring about chromosomal disorders like Down syndrome, for example. So if you're missing or have an extra chromosome, it causes various effects. Some are deadly, some are not. But all 23 are necessary. 23, they have different things on them, different genes on them. And you need all 23 to describe your whole body. There'll be two sets when they're copied. Not in your body, they're all the same set.
Wait.
Go ahead.
They're all the same set, but they're multiplied, right? I don't only have 23 chromosomes.
No. Every cell has a full set of 23. Yeah. So if there's a trillion cells in your body, then there's a trillion times 20. There's 23 trillion chromosomes in your body. Yeah. Right. Yeah, good question. So let's talk about the cell cycle. And this is kind of like, basically the cells in your body go through this cycle all the time. And it looks something like this. And your book has a picture like this on page 102. And it looks something like this. It's a pie chart. Okay? And this is the cycle. And it goes around like this. And this is basically, it's divided up like time, like how much time is it spending in each one. And there's one that is called the S phase. Okay? And in the S phase is where chromosomes are copied. And this is actually the one of the longer phases of the cell. Chromosomes are copied. And if you were to look in the cell at this point, you wouldn't see any chromosomes. The reason is they're just single strands of DNA and they're too small, they're too thin to see. Even with an electron microscope, you could not see them. Then we have, on the outside of this phase, we have what's called G1 and G2, and these are pretty simple. These are just called the gap phase. Basically, the cell is doing not too much. It's basically resting. That's why it's called a gap. Basically, if you're watching a cell under a microscope, something's happening here. Chromosomes are being copied even though you can't see them. And then something's going to happen over here. This is when the cell divides over here. But in the gaps, you wouldn't see, nothing's basically happening. The cell's just going about its normal business, making energy, digesting things, whatever it's normally doing. But it's not doing anything special getting ready for cell division. So chromosomes are copied in what's called the S phase. And then over here is what we're going to focus on today. This right here is called the M phase, which is also known as mitosis, which is cell division. Mitosis is cell division. And cell division excuse me, By the way, what I'm gonna do is I'm gonna put another layer around this circle like this. See what I'm doing here? Because, whoops, it's got to go all the way down to the G. These right here are called interphase. It's kind of like intermission. In between each division, there's a long intermission. In between each division, there's a long intermission called interphase. And that's G1, S, G2. Gap 1, replication stage, and then, or copying stage, and then gap 2. Now, what is mitosis? Mitosis has, I'm going to summarize it, but you're going to want to use your book probably for this activity. There's a nice picture on page 107 of mitosis. Looks like this. You see this? You're probably going to want to use this picture for the activity we're going to do next period. It's on page 107. There are 4 steps to mitosis. The first one is called, did you guys learn this before? In middle school or something? Prophase. Metaphase. And you have to memorize these, but fortunately there's only 4. Anaphase.
Why do they all have to sound exactly alike?
It's about job security for scientists. Like, if we can scare people away from science, then it will make less people enter science and make our jobs more secure. So, yeah, I'm sorry. I'm being sarcastic. I'm not supposed to do that. Prophase, metaphase, anaphase, and lastly, who remembers? Starts with a T. Telophase. Now, what I'm going to do is just highlight what's happening in each stage. And the cool thing about mitosis is you can actually observe this happening in the microscope, under a microscope. And we will look at some slides either tomorrow or Friday where you can see these different things happening. The reason you can see them happening is because in prophase, the chromosomes, sorry, I misspelled that. Now remember, a chromosome is just a coiled up, not a coiled up. A chromosome is a long strand of DNA that you can't see because it's too thin. It's a single molecule. It's only a few atoms thick. But what happens in prophase is the chromosomes coil up on themselves. Like an old telephone cord. Have you ever seen those old telephone cords? They're like a springy, like this. They coil up like this. And then they coil up again, and again, and again, and they coil up, I think, 3 times. Kind of folding up to the point where you can actually see them. And they look like this. Okay? And that is called a chromatid. Chromatid. Yeah.
What is God particle? Remember that?
That's different.
Yeah, I heard it.
Yeah, I don't know anything about it, actually. I know it's the Higgs boson, but you have to ask Dr. Ben. Okay, so this is a chromatid. Now listen. If you look back up here, you will realize that chromosomes have already been copied. Okay? So the second thing that happens in prophase is that you get the 2 copies joining up to each other like this. These are the 2 copies. All right, you stay with me now because this gets confusing. Every one of the 23 chromosomes got copied. Now you have 46. Every one of the 46 coils up and makes a short noodle-shaped thing. Okay? Now, the 2 copies, and don't ask me how they know they're copies, but the 2 copies come together and join to each other. Remember, somehow the cell has to keep track of the copies because it can't just randomly mix them up between the 2 daughters. It's got to keep track of them. This is how it does it. It puts the 2 copies together like a paper clip. And that little clip on the middle is called a centromere. The centromere joins the copies, okay? And the two copies are called sister chromatids. Okay, sister chromatids. When were the copies formed? They were formed before in S phase, not in mitosis. Copies were already here. What happened in prophase? They just coiled up, kind of like coagulated. And then they joined the copies together at the centromere. They're getting ready to keep track of, the cell's getting ready to keep track of all this stuff. There's one other thing that happens, and I didn't make myself enough room here. The centrioles form the spindle.
Spindle? That makes sense.
Yep, spindle. Now, some of you have seen centrioles before because I included it on a list before for one of the posters or something as a cell organelle, even though your book hasn't talked about it before now. Basically, a centriole is a little organ that works almost like a little pulley system. And the spindle is made of fibers, cytoskeleton fibers, that join like this. Okay? This is the spindle. These are the centrioles. That's the spindle. It's literally like, I guess it kind of looks like a basket. Like a fish trap, an old Native American fish trap. These are little fibers here. I don't know, I guess they're made of protein or something. And these are little like mechanical parts that attach. The fibers actually grow out from them and join in the middle. Pretty weird. This stuff is happening in a microscopic little cell. Now, in metaphase, when you think of metaphase, think of middle. Okay? Because what happens in metaphase is that the sister chromatids line up across the middle of the spindle. Like that. Yeah, it kind of looks like, except it's lined up differently, right? On the football, they go parallel to the football, right? Don't they? They line up across the spindle. Now, there was one other thing that was happening up here. The nuclear membrane breaks down, but I'll let you read about that. That's the basic idea of metaphase and anaphase. They start to move. I'm going to finish this up, this period, because in the next period we're going to do a little activity. Now look what's happening here. This is when they split up. So, you remember, I started this off by saying somehow the cell has to keep track of the copies and make sure that each daughter gets a full set. Here's how it does it. First, it joins the copies to each other. Every copy is paired with its copy. Then, it attaches a pair to each of 23 spindle fibers. There's 23 fibers. Each pair of copies gets attached to one. And then the spindle apparatus actually pulls apart the copies and moves them to opposite ends. So what will happen is those sister chromatids get separated and one copy moves to each side of this thing. It's literally going like chick, 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 and it's moving them down. It's like cranking those fibers down and moving the copies along to the other sides of the cells. Okay, now in telophase, we've got the separate copies on each side. The spindle goes away and new nuclear membrane starts to form. New nuclei form. Okay? then the last thing has to happen is that the cell is actually going to divide, and the cell cytokinesis is happening kind of at the same time. Technically, cytokinesis is not like one of the phases of mitosis. It starts happening, I don't know, in anaphase or something like that. But cytokinesis just means the actual splitting part. Mitosis, prophase, metaphase, anaphase, telophase. Those four phases are all about what's happening to the chromosomes. Okay? Cytokinesis is just the cell actually dividing. This is where it actually splits. Cyto meaning cell. It splits up the cytoplasm. And then you get two cells. So what happens is a little belt thing forms around the outside of the cell, and it just constricts and constricts and constricts until it splits the cell in two. Pinches it in the middle until it splits. And that is a brief overview of cell division. Now, when you get back,